Yeah.
Also, like,
Some of the things are so awful.
you know, I definitely find historically also like the sort of smaller things, like the harmonic resonance of those things, you know, this position or this
Yeah.
that's why I thought it was interesting.
Yeah. No, I get that.
Reasonable responsibility.
It is easy, but when it's so just like my experience of it's like all the
Yeah, right.
emails
Yeah, it's true.
mentoring me, all of the
Thanks again, Jim.
academics, and then I went to business research
So
and business tech search.
oh,
Oh, cool! I see that now. They're like, you will. You will see this. And continue to see this
no,
for
I
a
know
long
he's
time.
been busy. He won a contest once, and he's a lecturer in here. Yeah, no problem. The Lou
Yeah,
is
how's she doing?
a lot, right? Yeah. Yeah. Job security. But I still want to get the system updated.
Is there anything you could do?
I gotta check. I know
Yeah, I'm gonna be
I
on.
know personally, so I
The big heads involved. Yeah. Yeah, good luck with that. Oh boy. I have to get
Is she gonna get
don't worry,
offers?
I don't have to spend the money.
I
They
think so.
won't,
I
right?
mean,
That's what it seems like. Yeah.
she looks awesome.
But that's what it's telling us. Yeah, that's all we want to hear. You know it's bullshit. I know. I would say I'm not thinking. That's only what they're telling us.
you know, I would like to
Yeah, we should meet up.
garbage chips. It's not good.
Yeah.
I mean, it's better than salad.
What do you mean? It's great.
Um,
Well, to other people, because maybe I talked to Bravo, yeah, but he was not in confused.
yeah, and he explained nothing. It's great. My dad's just been pretty good.
Oh yeah, no,
Is that what your dad told you?
Well, my dad was a nurse navigator for many years. Bad job. Yeah, that's what brought him to DC.
Yeah.
And sort of like combination
the
of
news handles was awesome.
like this illusion of this dissatisfaction and kind of restlessness for better things and
No,
finding
he was not.
um, yeah,
Yeah, so I didn't meet him, so I'm not sure if he was not around.
and then finding
Oh yeah. I mean, I think it was good. Oh yeah, no. I know that. That is dangerous. That is truly dangerous. Yep. Yep.
It turns back like every you find is very best use of your time because I've tried that too. Some talks where it's like, oh, in the first 5 minutes I'm very effective, but then after 2 years, I mean, I move through different things. Sounds better. I'm actually honestly.
Yeah, I haven't.
Hello, everyone. It's my great pleasure to introduce Patricia Janak for today's seminar. She's a Bloomberg Distinguished Professor at Johns Hopkins University Psychology Department and also Department of Neurobiology. She's a real pioneer of using optogenetics in behavior experiments. She's one of the earliest users of optogenetics in behavior experiments. But also I would like to mention that she really distinguishes from other people by introducing very important classic animal learning theories as well as modern behavioral neuroscience frameworks. So that really makes her work impactful compared to other works. And to do that, she graduated from Rutgers College, majoring biology and psychology. And then she did her graduate work at UC Berkeley. She started studying addiction, including cocaine and alcohol, which remains her main topics these days as well. And then she got a faculty position. So after a couple transitions, she got a faculty position at University of California, San Francisco, UCSF, and then she remained there for some time. And then in 2014, she moved to Johns Hopkins, and that's where she is currently. So I was very excited to have her now, because now I think the big question in the field is how to understand the heterogeneity of dopamine neurons. The classic view that dopamine neurons are just sending reward prediction error has eroded already. So I think the next questions are really try to figure out what they actually do, and I think her work gives one of the most important insights on this topic. So I'm particularly excited to have her here. So please.
Thank you so much for that wonderful introduction. It's a real pleasure to be here. Is this mic working now? Yep. Okay, good. Got a thumbs up. It's a pleasure to be here. I've had some wonderful meetings so far this morning, and I really look forward to the rest of the day. So as now mentioned, I'm going to be talking to you about our studies looking at the function of dopamine, focusing primarily on learning. So our lab is very interested in reward learning in general, both how we learn to acquire, natural rewards, if you want to call this a natural reward, how we learned what the symbols mean, how we learned that this black liquid is something, that we like the taste of. And we're also interested in understanding that in the context of drugs. So a lot of our work is looking at learning processes that underlie acquisition of alcohol intake or cocaine intake, and more recently opioid intake. But today I just want to talk about some studies that I view as really actually incredibly almost simple-minded, but I think I hope have given us some nice ideas about how dopamine functions in reward learning. So this is one of these classic cartoons of the so-called reward circuitry in the brain. This is from a review by Paul Kenny that highlights some of the areas of interest like the amygdala and the OFC and the striatum, but especially these dopaminergic regions, the substantia nigra and the ventral tegmental area in the midbrain. And to think about how dopamine may participate in reward learning, we're going to break down this very complex process into the most simple units that we can think of, and those would be Pavlovian conditioning and instrumental conditioning, these very simple associations between cues and rewards they predict, or actions and rewards that follow those actions. And we're going to think about how dopamine may act in acquisition of these kinds of associations. Of course, as you all know, the primary guiding hypothesis for dopamine's role in learning is that it acts as a reward prediction error. And this kind of slide people have probably seen too many times and may be sick of, but I'll still say it quickly in case there are some new students who aren't yet sick of it. I'm not sick of it. I still think it's beautiful. And as you know, the idea that dopamine firing can act as some kind of reward prediction error comes from the close match between the way dopamine neurons respond to reward, whether it's surprising or unexpected, fully predicted or omitted when expected, and the direction of the response parallels a signed prediction error that comes from early learning models in psychology, like the Rescorla-Wagner model, or more modern TD models used in computer science, right? So the way the neurons react to reward is not fixed, but is dynamic depending on the animal's expectation. Surprising reward is a brief increase in activity, expected reward, little change, omitted, a depression. And so this was in the monkey, classical work from Schultz and colleagues, and in the years that followed, people turned to rodents, and you can see cue and reward signals just like canonical monkey signals in the rodent, including the rat and including the mouse. And I mention this always, I try to when I make this longer talk, because this piece of work out of the Uchida lab was really key, because for the first time, using optogenetics, we were able to say that it truly is identified dopamine neurons, at least a large subset of them, that can show this classic RPE sort of profile. And so here's just one of the figures from the paper showing increase in activity for something positive, the reward, a decrease in activity to omitted reward. So there were all of these lovely EFIS studies showing a nice correlation between the activity of dopamine neurons and reward prediction errors in conditions when animals were learning or were changing their associations with cues and rewards. And there also were plenty of pharmacological studies using drugs to change dopamine in the brain and also showing an association with learning. But we wanted to try to make a more direct causal link between dopamine neuron activity, prediction errors, and learning. And so we thought a few years back that we could take advantage of optogenetics. When we first thought of this, it was just emerging upon the scene. And so we were very excited by the opportunity to use optogenetics to produce artificial positive prediction errors and artificial negative prediction errors and test whether we could change behavior the way theories would predict. So when we began this work back at UCSF, we first chose for a behavioral procedure in which to address this question the classic behavioral task used in animal learning to show that prediction errors are important for learning. Just the concept of a prediction error is important for learning. And this is the blocking task. And I'll talk about this for the next few slides, and that's the most complicated behavioral task. Everything else is super simple for people that are not into behavioral tasks. Okay, so in the blocking task, you have your group of animals, in this case a group of rats, and they have two phases of conditioning. 2 phases of training. In the first phase, you play a cue, let's say it's a sound, and you follow it by a nice large sucrose reward. That's what this symbolizes. And you repeat this pairing till the animal learns that the cue predicts the availability of reward. After the completion of phase 1, you move to phase 2. And the question that was asked quite a long time ago by Kamen and others in the animal learning field is, if you now added another cue, represented here by X, in our experiment, it's going to be a visual cue, a light. If you added another cue and played it at the same time as X, still followed it by the same reward, what would the animal learn about this new cue X? And so later on, you could test that by just presenting X and looking at the animal's behavior. Did he show any conditioned responses? And you can compare what happens in this group of animals with a control group where critically the first cue they learn about is not a cue they see in this second phase. And so why is that critical? It's critical because in this second phase of learning, when we add new cues, in the first blocking group, when X is added to A already fully predicts the reward. We've not changed A, we've not changed the amount of reward the animal gets, so there's no prediction error occurring here. So if prediction errors drive learning, then you wouldn't expect to observe much conditioned responding to X at test. Alternatively, here, when you get to this second phase, both A and X are new cues that have never been followed by reward as far as these animals in the control group are concerned. So there is a positive prediction error, and you should drive learning to A and X. The animal should learn, aha, here's a new set of cues that mean this reward. I need to learn about them, and you should be able to see more behavior to X in this condition than blocking. This has been shown many, many times in many different kinds of situations across species, including humans, and in our replication of this in rats, this is what this kind of effect looks like. If you just look here at the right, the difference between the response to X in the blocked and control group. You have more responding in the control group that presumably displayed a prediction error. So why am I going through all this? It's because this procedure allows us to test whether VTA dopamine neuron activity at the time of reward can promote learning about preceding cues. And so, as I mentioned, we'll do that using optogenetics, which you all already understand and know is used to control the activity of neurons using laser. So it puts dopamine neuron activity under the control of the experimenters. And we'll ask if, in a situation where there normally is no prediction error, when no learning would naturally occur, if we activate VTA dopamine neurons at the time of expected reward, we should generate an artificial positive prediction error and promote some learning. So what we're trying to do is to unblock learning to X. So this experiment was really the brainchild of Ronald Keiflin, who's now an assistant professor at UC Santa Barbara, and he was a postdoc in the lab at this time, and he worked together with Liz Steinberg, who's now a postdoc in the Malenka lab, to use the TH-Cre rat in order to test this hypothesis. So they expressed channelrhodopsin unilaterally in dopamine neurons in the ventral tegmental area of TH-Cre rats and performed the following experiment. Using this same blocking behavioral design, remember in our traditional group we have a cue that's predictive of reward. After this is learned, you add a second cue that's played at the same time. So this is a compound cue, still followed by the same reward. Normally, little learning, little associative strength would accrue to X. We're going to artificially activate dopamine neurons at the time of reward delivery to drive an artificial RPE and see if we can get learning to X. Of course, we need control groups. The control groups are Cre-negative animals. They don't express channelrhodopsin, so the dopamine neurons won't fire. And the critical control is the unpaired Cre-positive animals, where we will stimulate dopamine neurons, but not paired with the reward or the cue, by the way. It will be in the ITI. Because the critical aspect of the hypothesis is that the timing of when the dopamine neurons fire needs to be during the time, that reward period, during the time that reward occurs. And so to make a long story short, when we ran these groups, we indeed found evidence for unblocking by dopamine neuron activation. So an artificial reward prediction error, if you turn here to the right, results in more responding to QX at test in this group, our experimental group, compared to our two control groups. And you can see this here in this test run in extinction, where you just briefly present the cue. The animal shows that it knows what the cue means because it enters the port looking for the reward. And that's what we see right here. So we were really excited by these findings. because they supported the idea that activation of dopamine neurons after a cue can drive learning about that cue. They supported the idea that dopamine neuron activity can act, at least in this condition, as a reward prediction error. And that's what I'm saying right here. So moving on from this result, we wondered if our findings resembled natural unblocking. Unblocking is a real occurrence that happens when reward size changes or when reward identity changes. Normally that produces a surprise. It's a surprise to the animal. It produces a reward prediction error and can drive learning. So Ronald went on to replicate and extend these results by comparing unblocking by activation of dopamine neurons with unblocking by a natural change in the magnitude of reward. So we've got the same kind of letters, etc., that can sometimes drive people bonkers, but now we have a within subject study where each subject in the initial period learns about 2 cues, and each cue is predictive of reward. In the natural reward upshift case, this cue is predictive of large reward, B of small reward. The critical time period is this compound cue phase. In the natural reward condition, when we get to the compound cue phase, we add a second cue that's played at the same time as A and that's played at the same time as B. One's a sound, one's a visual cue. And then we follow by reward. In this case, it's the same amount of reward. This is the classic blocked condition where animals don't show later learning to X. However, here in this case, we've increased the magnitude of the reward. So the animal's getting B and Y and a bigger reward that it did not expect. There's a reward prediction error. We expect to see increased responding to Y in this case compared to X at test. We can compare that with the same kind of procedure when we use activation of dopamine neurons to give this fake RPE. So in this kind of case, we want to hold the real reward constant, and we just add our fake RPE by turning on the laser and activating VTA dopamine neurons. And so if this condition is similar to this, we would expect to see unblocking in both cases to Y and blocking to X. So greater responding to Y than X. Ronald was also interested in whether this response, this ability of dopamine neurons to unblock learning and drive learning to cues, was specific to the VTA, the first dopamine cell group he looked at, or if it would also be shared by the other major midbrain dopamine group, the substantia nigra pars compacta. So this was a really giant study where he had 3 very big groups trained in this kind of procedure where they have 2 cues, 2 sort of cue lines that they're trained in this within subject design. So some animals received channelrhodopsin in their VTA and others in their substantia nigra. And so the artificial reward prediction error was produced by shining light either into the VTA or the substantia nigra. And then everybody's trained the same. And we're going to look out here on the right and compare responses to X and Y. And first of all, see if we see unblocking from natural reward upshift. And we want to know if we replicate the effect of stimulating the VTA. And we want to know if we will also see that after stimulating the substantia nigra. And here's just the acquisition curves of all of the animals. The critical data to look at are here on the very far right, and it's the responding to QX versus QY. Here's the natural reward upshift group. We see an increase in responding to the unblocked cue. So this is a classic unblocking effect. You change the magnitude of the reward, they learn more about the new second cue. And it looks almost identical to what we see here with the VTA, which was really exciting to us. So we feel like we are able to capture and mimic the natural kind of updating that occurs when reward magnitude changes by stimulating VTA dopamine neurons. So this difference between X and Y here, this is our VTA group. In contrast, in the substantia nigra group, we see no unblocking. So in this particular procedure where we're driving responding to cues, VTA dopamine neuron stimulation works, substantia nigra neuron stimulation does not work. This was interesting to us because if you were Wolfram Schultz recording from neurons, when monkeys see cues paired with reward, you see reward and cue responses throughout all of the dopamine neurons. This suggests that they have different functions specifically, possibly. Ronald went on to try to understand a little bit more about the kind of learning happening in this VTA group after unblocking. So what was it the animals were learning about why? And I'll just make sort of a quick digression to talk about this, and I'm happy to talk about this more later if people are interested. What he wanted to know is whether animals just have increased the value of the cue, or whether they actually are understanding something about QY and its relationship with sucrose reward, the identity of that reward, specific aspects of that reward. And this is the kind of distinction that people talk about when they talk about model-free versus model-based learning. And historically, dopamine, acting as a reward prediction error, has been thought to drive a sort of model-free, incremental kind of learning where you adjust the value of actions and cues without really adjusting the information that the animal has, the richer level of information that the animal has about, for example, the nature of the specific reward. Whereas model-based, and these are my very crude definitions of model-free and model-based. I'm sure there are much, much better definitions that we could use. But in model-based learning, it involves more complexity and actually understanding the details of the current environment and what is learned, exactly what reward a given cue predicts, for example. So the classic way to test whether an animal is able to use the actual representation of the reward to guide his behavior, model-based, or is just responding in a model-free way to the value of the cue, is to devalue the reward. And so what Ronald did was repeat this same kind of training in this blocking experiment for VTA animals. And he took half of the animals after training, so everyone's trained the same. Half of the animals, he devalued the sucrose reward through conditioned taste aversion. So he paired the sucrose reward with the lithium chloride injection. This is a standard way to reduce the value of a reward. It's done outside the conditioning chamber in the home cage. And there's a control group called valued. They receive lithium chloride, but it's not paired with sucrose reward. And what he's going to ask is whether changing the value of the sucrose will alter the way animals respond to the cue they just learned about from the opto stimulation. So will it alter their responding to Y? So if this ability to drive learning to Y here is really just reflecting a model-free process, you would expect no difference in the responding at test between these two groups. And that's what we expected. We thought, dopamine is just going to be acting in this model-free way, updating the value of the cue, but the animal won't necessarily connect that with the specific reward that cue predicts. And in fact, Ronald saw the opposite. So here you can see at test the way that these two groups of animals responded to QY. And you can see the devalued animals for whom the value of sucrose was reduced by CTA. They respond much lower at test. They're seeking the sucrose in the port much less than the valued animals who respond just the same as in the prior experiments I've already showed you. So this was a very surprising finding that was interesting to us, and it suggests that even when we use this artificial way to activate dopamine neurons, that in some cases might be acting as a model-free sort of incremental learning signal, we can still potentially intersect with more complicated learning systems in the brain, like model-based learning systems. So we've not followed this up yet to try to understand how that actually might be implemented, and it's something we look forward to doing. But the overall conclusion is not only can VTA stimulation drive learning about the cue, but at least in this condition, it can drive this more richer learning where animals are able to associate cues with actual rich details about particular rewards. Mm-hmm.
So that means the sucrose solution that they got in the cage kind of is generalized to the sucrose solution they got in the training chamber.
That's right. Yeah. So if you
There's an interval between this, the test training and switching from model.
So as far as I know, for conditioned taste aversion, it doesn't matter, at least within the range that people typically use in experiments, because CTA is very strong. And at least in this case, for this experiment, once we did the CTA, it was very long-lasting. If we had devalued by pre-feeding, it's very important. So pre-feeding, you just devalue by temporarily satiating animals on a certain taste. that will be gone in a few hours. So that, in that case, it's very important. And it would have been interesting if we had done that and gone back and forth in this condition, but we did the hammer approach with the CTA and really drove down the value of the sucrose. So in these initial studies, I've shown you some of our evidence that activity at the time of reward delivery can promote learning about antecedent events. And while we were conducting these studies, there were a series of really beautiful studies also using optogenetics to manipulate dopamine neurons coming out of the Schoenbaum lab and other labs also using this approach to try to make causal connections between positive and negative prediction errors and learning. So overall, I think we have pretty good evidence. You can mess with dopamine neurons on a short time scale with optogenetics and drive learning or about things that happen right before you do that. These conditions have typically been Pavlovian, and so we still were interested in whether we could think more about possible differences in the way dopamine might contribute to Pavlovian associations and instrumental associations. And we were still wondering, what about the substantia nigra? We didn't see an effect of activating substantia nigra in our blocking procedure, which is a Pavlovian procedure. What is that dopamine signal for, that brief burst of dopamine neuron activity? Yes?
Why do you think the animals don't sort of associate X with A? Why aren't they making that association and just having a second order conditioning where now X should also predict A and therefore they should vote for reward?
Just because it's at the same time, yeah. And so this literature, I know there are other people, I wish Ronald was here because he could recall the years and years of literature, but you can do different kinds of tests to basically see how much value each of the individual cues get to them when you split them up in different sorts of more complicated things like that, that seem to suggest that they're viewing them really as a compound. So in the next group of studies, we wanted to more closely compare substantia nigra and ventral tegmental area, and then also compare what's going on in these two really, really simple canonical kinds of associations that underlie so much of our reward-seeking behavior. And what I didn't mention before, but which you already know, we're interested in the VTA versus the substantia nigra because, of course, they have different targets. Right, the striatum is a very large structure, and you know from lots of beautiful work done in this department that there are very distinct projections from midbrain dopamine neurons to targets, including the striatum. This is a vast oversimplification, but we can think of the ventral tegmental area as projecting primarily to ventral striatum or nucleus accumbens, substantia nigra in the experiments I'll talk about, really talking about the more lateral substantia nigra projecting mostly to dorsolateral striatum. And we'd like to do experiments using optogenetics to activate one group of neurons versus the other. But this diagram, it makes the key point that these experiments that I'm going to tell you about are not clean in the sense that these are not separate cortical striatal circuits, but there are lots of ways that there can be crosstalk between them. And one of the most sort of currently famous ways is through an anatomical relationship originally described by Suzanne Haber in the primate that is unidirectional. It's a spiral moving from VTA to the more lateral substantia nigra areas. And it's based on the finding that dopamine neurons can project to a certain area of the striatum, for example, in the ventral striatum, and those medium spiny neurons in part project back to a different subterritory in the midbrain, and through an intermediary GABA neuron impact the next set of dopamine projections, and so on and so on. So you have this spiraling connectivity. And there are good pieces of evidence in the literature that behaviorally this may be important, but it's something that people are still trying to piece together. And it's something we need to keep in mind for the next experiments I'll talk about. So we have these super basic associations, Pavlovian conditioning and instrumental conditioning. And the idea with dopamine reward prediction error would be when you or an animal are first learning that a cue is followed by a given reward, it should be a surprising event. And the first time you make an action and it's followed by a given reward, it should be a surprising event. So your dopamine neurons should signal something. They should fire. And so what we decided to do was really reduce the complexity of this situation and remove the actual reward and just look at a situation where we pair cues with dopamine neuron activation and pair actions with dopamine neuron activation. So we're just trying to when you actually ingest reward, all kinds of things are happening in the brain, all kinds of interesting, cool processes. We're eliminating those and just going for the neural effect on the dopamine neuron system. And so this first set of experiments looking at Pavlovian conditioning was conducted by Ben Saunders when he was a postdoc in the lab. He's now an assistant professor at the University of Minnesota. And what he did was take THC rats and infuse virus to express channel rhodopsin in either the VTA or the substantia nigra unilaterally, implant an optical fiber, and then when the time was right, take his rats, put them in a chamber they've never been in before, play a cue, it's a sound and a light on the wall, and turn on the cue, and after the cue's been on for a couple of seconds, to in fact turn on a laser and activate dopamine neurons at 20 hertz for 5 seconds. And he did this, I think it's 16 times a day for 14 days, so multiple trials per day for many days. There is no food available here, no drug. He's videotaping what the animal does. And so what he did then was look at the videotapes and see what is the behavior of the animals when they receive paired cues and dopamine neuron stimulation. Of course, there are always unpaired control groups where the cues and the stimulation are not correlated in time. And using your standard sort of video tracking software, he observed that some changes in behavior occurred at the time the cue went on over days. So if you look at, for example, at velocity for the two groups at the final day of training, day 12, you can see data here from purple, those are substantia nigra animals, and blue, those are VTA animals. At time 0, when the cue comes on, there is basically a conditioned locomotion response, right? You see an increase in activity to the cue before optogenetic activation of dopamine neurons. So this suggests that there's a change in behavior. You've promoted, by pairing cues and activation, some sort of change in behavior. Critically, this does not happen in unpaired animals. So just repeatedly turning the opto on, you're not causing them to move, you're not sensitizing their movement. So this depended on pairing of a sensory event with activation of dopamine neurons. We're trying to mimic natural Pavlovian conditioning, where cues are followed by reward. If we've really mimicked it, then we should expect to see in the dopamine neurons the appearance of a response to that cue. So one question that Ben asked was, if he looked at dopamine neurons in the VTA and substantia nigra, would he now see a response to the cue that was now eliciting movement? And so to do this, he conducted the same kind of training experiment where animals get cues followed by optogenetic activation, and he combined it with in vivo photometry to measure changes in bulk calcium activity in dopamine neurons in the midbrain. So he had to switch to crimson so that he could activate the neurons and still record the calcium activity. And so when he did that and looked at what happened at the time of cue onset from the first session to the last session, you see a really clear increase in the magnitude of the response to the cue and the duration of the response to the cue. So blue is the response to the cue that develops over the first session. Orange is the response to the cue on day 12. So this is the classic increase in cue response you would expect to see over learning. The other lines are the unpaired controls. There's nothing happening there. So just as you would expect for natural reward conditioning, photometry shows you a cue response in those dopamine neurons, and it's similar in magnitude to natural reward learning. We've done that parallel control experiment. So this was good. This made us feel like, okay, we are mimicking at least some aspect of the kind of normal conditioning process that happens for dopamine neurons. So let's think back now more to the animal's actual behavior. So I showed you an increase in velocity we got from these numbers that came out of the tracking program, but when you look at the animals, what are they actually doing? And so I want to show you one trial on a VTA animal, if I can get the mouse, here it comes, this is day 12, he's well trained. You'll see the light come on. It'll be on for 2 seconds before the optogenetic activation happens, and he has a very, simple yet prototypical behavioral response. The light comes on, he approaches the light, he sniffs it, he attends to it. So this is cue approach. And anyone who's worked with rats in any kind of cued reward paradigm knows that a good portion of the time they'll spend interacting with the actual cue rather than just the reward. This is also known as sign tracking. So when you look across animals at the group data, for animals who had their VTA dopamine neurons stimulated after a cue, you see an increase in the probability of approaching the cue on a given trial over training. However, you don't see this at all in substantia nigra animals. So looking at the videos, scoring those videos, there's no perceptible change in responding to the cue. In fact, they don't tend to approach the cue at all. So we have already a difference between the kind of behavior animals are emitting with this very simple procedure that we subjected them to. So what are substantia nigra animals doing? So remember, this was a unilateral manipulation. So we're stimulating dopamine neurons on one side of the brain repeatedly following a cue in substantia nigra animals. And now you'll see one trial from this guy, and again the cue will come on, and then you'll see his behavior. The cue comes on, contralateral circling. No circling is observed in the unpaired control animals. It's not an unconditioned obligatory effect of just driving dopamine neurons repeatedly. It's somehow gated by pairing with the cue. So if you look across the group of animals who received activation of their substantia nigra, you see a really giant increase in this rotation behavior over the 12 days. However, things don't stay simple, because VTA-stimulated animals also begin to show this rotation behavior over the 12 days. And I'm showing that here in this graph, and the rotation behavior is in this lighter blue color for VTA animals. You can see they start much later and don't reach the same extent as fast as these animals. So they really show a mix of approach and rotation behavior, sometimes mixed on a given trial, sometimes one trial one thing, one trial another, but it clearly is changing over days. So we don't have a clean dissection of the two regions, and so this is one area of the results where we think, but we don't yet know, of course, that the reason why VTA animals start to rotate is because by repeatedly activating these neurons following cues, we're over time recruiting these more lateral circuits and potentially getting down over to the substantia nigra and recruiting whatever this process is that's mediated by activation of the substantia nigra after a cue that we see in these animals. We don't see approach in these animals because this recruitment is theoretically one way. And so this is an idea that we're in the middle of testing. I'd love to hear other possibilities if people have other ideas.
But why circling?
Because it,
Rather than any type of increased locomotion?
because it's unilateral. So that's a very important question. So we would predict that if we had done this bilaterally, it would be some other kind of conditioned locomotion, and that actually would be an important control study. So it's because of the contralateral effect. So we imagine that the plasticity that we're engendering is more on one side, probably, in a perfect world, it's corticostriatal plasticity. We're impacting more on one side than the other. So because the cue that was paired with VTA dopamine neuron stimulation engenders approach, we can think about that effect in light of a whole set of classic behavioral studies people have made trying to understand how cues can come to be incentives, how they can motivate action by animals. And the idea that people have come up with over the years is that cues paired with reward acquire value in their own right. So they acquire some kind of value such that they attract attention and cause approach. And you can also measure the value they've acquired by looking to see if they can support learning a new behavior. And that's called conditioned reward or conditioned reinforcement. And so that's the second classic test about whether or not a cue has acquired rewarding value. And so Ben went on to conduct that test in this experiment. And so in a conditioned reinforcing experiment, you test, after the pairing of a cue with reward, you test whether that cue will serve as a reward itself. And the way to do that is through a two-phase experiment where you first have the pairing. That's the same conditioning we've already been talking about. In fact, the same animals I've already shown you. And then you have a second phase where you now put them in the chamber and you ask them to learn to lever press, but instead of getting a reward or dopamine neuron stimulation, all they get is the cue. So you just turn on the cue for a couple of seconds. There are two levers. If the cue has acquired some reward and can support the acquisition of a new response, animals will work more for that cue. They'll press that lever more than a control lever that does nothing. So when you look at the VTA animals versus substantia nigra animals, you indeed see that VTA-trained animals do show actually really strong conditioned reward responding. They'll respond quite a bit for the cue that previously was paired with dopamine neuron stimulation. Substantia nigra animals will not do this at all. So this is a second piece of evidence that when we do this sort of Pavlovian conditioning procedure, that the particular contribution made by VTA dopamine is that it imbues cues with some kind of value. So people will call this incentive motivation value, for example, because it comes to control the animal's behavior or motivate his behavior. So the cue motivates specific behaviors directed towards the cue. In contrast, for the substantia nigra situation, so far all we can say is that it induces some kind of general motor invigoration after pairing of cue with dopamine neuron stimulation. It's not focused towards the cue. There's nothing else for it to be focused to in the cage. There's no real reward. So this is something that we're very interested in following up to understand the relationship between the increase in motor activity and this Pavlovian conditioning. So far we've been talking about very crude manipulations of VTA dopamine neurons and substantia nigra dopamine neurons without regard to the exact striatal territories they're projecting to. And so we have begun to try to refine that by using retrogradely transported viruses that can express channel rhodopsin. In this particular study, that injected virus into the nucleus accumbens core or shell and into the dorsolateral striatum and repeated the experiment and looked to see if he could recapitulate the effects of VTA stimulation and substantia nigra stimulation. And the results for the substantia nigra to DLS projection were exactly the same. There was contralateral circling, no cue approach, no conditioned reward. And for the dopamine neurons projecting to the nucleus accumbens shell, there also was no cue approach and no conditioned reward. And it was only dopamine neurons that project from the VTA to the nucleus accumbens core that fully recapitulated the approach to the cue and the conditioned rewarding properties of the cue. So this matches prior work on conditioned rewarding properties of cues that has been done over the years from Barry Everett and Trevor Robbins and many, many other labs. So it's specifically dopamine neurons projecting to the core that seem important for this assigning value to cues in this way. So that's where we had gotten as far as looking at Pavlovian associations and looking at VTA versus substantia nigra. We want to also address what the similarities or differences might be in the realm of instrumental conditioning, where animals are pressing levers and now in this case getting activation of their dopamine neurons. And of course, this is simply intracranial self-stimulation, which you probably know. Animals will work to activate their own dopamine neurons, whether it's substantia nigra or VTA. But when Ronald Keiflin looked really closely at their pattern of responding, when they're over many, many hours, you can see very clearly VTA animals will just keep working with and hardly take any breaks at all for hours and hours and hours. Whereas for substantia nigra, animals will press levers to activate their substantia nigra dopamine neurons, but there are all these very long breaks. And when he quantified this, the breaks, it's quite different. That quantification is here. And so Ronald tried to begin to think, why would this be? You would think that it's just a ICSS. Why isn't it the same? Why is it different? And in thinking about it in light of ideas about VTA versus substantia nigra, and in light of the data I just showed you, he came up with a framework to try to think about these and some new tests for the instrumental learning. And the idea would be that dopamine signals from the VTA can support animals learning about the value of cues. That's what I just told you. And in the case of ICSS, the animal has to learn about the value of the different cues in the environment when you put him in the chamber. He has to learn to recognize the lever and to approach the lever. This is the lever that he presses to get activation of his dopamine neurons. If this is a function mediated by VTA dopamine neurons, then animals who are lever pressing just to receive activation of substantia nigra dopamine neurons aren't getting this. So they may not be getting the part of reward learning that includes understanding the cues in your environment and learning to to approach the correct reward signaling cues. And so that would mean they should be less efficient at getting stimulation from the lever. And if they disengage and turn around, they wouldn't necessarily immediately recognize the lever as something to speed back to get stimulation. So that's a sort of a simple way that we've been thinking about this. And so in designing tests to address this, this is basically the same thing I just said. Ronald first thought, okay, if substantia nigra animals have trouble coming back to the lever after they leave it, let's force them to leave it and see if that's true. So he designed an experiment where the lever is out. When the animal presses the lever, he gets activation of his dopamine neurons, and then the lever retracts for 12 seconds before it comes back out. So for the 12 seconds, the animal stops pressing, he moves away. The idea would be substantia nigra animals would have more trouble continuing to lever press in that condition. VTA animals would have no trouble because they understand the lever as a signal for reward. And in fact, that's exactly what he saw. So in these graphs that I'm showing you, you see regular ICSS when the lever is out all the time. That's no time out. And purple is responding by substantia nigra animals, and blue is responding by VTA animals, and they both work quite a bit over 2 hours to get stimulation of their dopamine neurons. But now when you change the session to be forced timeout, you can see responding certainly drops. The number of responses drops for VTA animals because the lever is gone quite a bit of the time, but it goes to 0 for substantia nigra animals. They really can't do it. And if you analyze response rate for the time when the lever is out, it actually increases for VTA animals. They're optimizing their ability to get stimulation, and it's still 0. So they can't do this. So that supports this sort of general idea. Another experiment that Ronald did, well, he tried to think, how can I change the experiment to try to make a situation where an animal would have to be flexible and learn how to recognize what different stimuli mean in the environment. And so one simple thing he decided to do was just to change the location of the lever after an animal had learned to lever press it for activation of his dopamine neurons. And so when he does this experiment for VTA and substantia nigra animals, all animals press a little bit less when you suddenly move the lever up 5 inches, but substantia nigra animals have a bigger reduction. So here in this purplish color, this is responding by VTA animals at the standard location. Then you move the location of the lever. VTA animals do begin to adapt. They're responding slower, but still pretty good. Substantia nigra animals start off the same, but they don't adapt as quickly. So this is another piece of evidence that goes along with this same kind of idea. And we're actually in the middle of trying all these variations to try to probe how the animal is using the information in the cage. So I thought about turning the lever upside down, and I'd love to hear other fun ideas that people can think of that we can do with the equipment in our boxes to further probe this. The third way that Ronald thought to test this idea that there are different kinds of learning supported by the VTA than the substantia nigra when animals are learning to make actions for reward, in this case being the activation of their own dopamine neurons, was to test their behavior in the acquisition of a response chain. So in this case, for Ronald, the response chain involved animals learning to make 3 different actions, and the last action is reinforced by activation of their dopamine neurons. But they learn this in stages. So first you learn the very first simple action, where you learn to press a lever for activation of your dopamine neurons. That's ICSS. After 3 days of training here, he retracts that original lever, and there's a new lever out in the cage. The animal has to press the new lever for access to the old lever to get the reward. After 3 days at this chain length of 2, animals have to nose poke on a different wall for the second lever to come out. When the second lever comes out, he has to press that to get access to the final lever. So learning how to chain these responses would require the animal understanding what the potential value to be gained by that action is at each of these different states. He would have to understand what each of these levers, in a sense, stands for and means. And what we are proposing is that substantia nigra dopamine learning processes alone cannot fully account for that kind of learning that would be necessary. So when he trained animals in this procedure, he sees, here are some just responses of individual animals. Each line is an individual animal. You see that at the first stage, everybody can pretty well do it. Blue is VTA, purple is substantia nigra. But as soon as you move to this middle stage, where an animal has to do one action to get access to the last lever, substantia nigra animals fall off completely. There's one animal that can do it, but none of the others can do that. VTA animals, it's hard for them, but they can certainly do it. And then when you move to the final phase, for the final 3 days of this 9-day training, only VTA animals can learn to make a nose poke for this lever in order to get access to this lever. And this is the first set of data. This has since been replicated with a larger group of animals, including animals from the substantia nigra group that are higher up and animals from the VTA that are lower up. So it's not just a quantitative difference, but it's truly a qualitative difference. The substantia nigra, at least in the place that we're stimulating the substantia nigra, we're trying to be quite lateral, cannot support the animals learning a chain of actions. Only the VTA can do this, at least so far in our hands. And this is just the group data. So this is consistent with the idea that maybe the dopamine from the VTA, these kinds of phasic learning signals, are helping the animal learn about Pavlovian state values, and that maybe the phasic dopamine activity in the substantia nigra, at least the area that we're looking in, perhaps is more concerned just with action value, just the action. It can only reinforce the immediately preceding action. And so we don't know yet if it really is qualitatively different like that, or if instead there is just a difference in the time over which these signals might act. That's something we need to check as well. But the idea is that when we're activating VTA dopamine neurons in these super simple learning procedures, we're able to engage whatever the natural learning process is by this circuit, and then also through recruitment, able to engage these other natural learning processes. So animals get the whole deal, right? They can learn the meaning of cues, they can have individual actions reinforced because we're able to activate this whole circuit. That would be a hypothesis that we're in the middle of testing, so not sure if that's true, but that's what the hypothesis we're working under. In contrast, when we use optogenetics to just come in here, at this point in the circuit, we don't have access to these other circuits in this spiral, and we're only able to activate whatever learning process this part of this spiral is important for. And at least so far, it looks like it's really only able to help animals acquire actions, very specific actions. And to be able to organize those actions, or even to recognize entirely when to do those actions, we would propose requires some other functions by these other preceding circuits. So to summarize what I've tried to tell you using just this one simple method of crudely activating groups of dopamine neurons, is if you do that during the time that animals get reward or expect reward or used to get reward, you can control subsequent responding for that reward, and you can control subsequent responding to cues that predict that reward. And so activation in lieu of reward can drive conditioned responses to cues, so you don't need the actual reward. And dopamine neuron activation after an action can increase the likelihood of that. And of course, these effects depend on which dopamine projection we activated. And so what we would like to say is that this is more evidence for something we already knew, right? Which is that different corticostriatal circuits control different kinds of behavior. This is something that's been known for a long time from all kinds of elegant studies from humans down to rodents. But here we're trying to understand how this seemingly similar function of dopamine neurons firing at unexpected rewards affects different classes of behavior based on where those dopamine neurons are projecting. And so we would say that at least in the experiments that we talked about today, dopamine is acting as some sort of reward prediction error to change behavior, but the behavior that's changed depends on the region to which those dopamine neurons project. And in the most elemental form, one thing that's fascinating to me is that when there's no real reward present at all, you're just activating dopamine neurons, you can still engage this system and reveal learned responses, conditioned responses to cues. So at the most fundamental level, what dopamine is doing in striatal circuits doesn't depend on some sort of deep knowledge about the nature of the reward or the identity of the reward. That doesn't mean to say that it doesn't contribute when you're in a richer learning situation and real reward is actually present. And so that we showed in the condition with the devaluation experiment, when the animals actually had real sucrose there, they actually were able to use the dopamine signal to form a richer association. And this is basically the same thing I already said. So VTA dopamine neuron activity is important for subjects to learn the motivational significance of cues. This doesn't require identity of the reward. It's really about how the organism reacts to sensory stimuli in the environment. Of course, in the real world, knowing about the identity of the reward is very important. When you actually ingest a real reward, many different circuits in the brain are activated, right, that interact with these same dopamine systems, and it's a much richer learning system. And so I've tried to tell you just a little bit about our work on dopamine's role in learning, and we hope that by following this up and also looking at this in the setting of drug addiction and drug intake, we can understand more about how these changes happen in the brain to support these both normal and abnormal reward-seeking behaviors in the future. I think I mentioned the lab members that participated most importantly in these studies as we went on, but they especially include Ben and Ronald, who now are in their independent PI positions. And I want to thank my funding and my collaborators, and thank you for listening to this slightly overlong. Thank you. Hi.
So I have three questions. One and two are related. So the sequential action. So I think animals brain needs to train also the action policies, and they need SNC dopamine. In your model, you need SNC dopamine. But do you think in this case the BTA, because of the interaction of the BTA dopamine, SNC dopamine, that's just overcome that, or the for this specific behavior, BTA dopamine be enough?
That's exactly what we want to know. So what I would like to think is that by activating the VTA, we're also recruiting substantia nigra systems, and that they are also necessary potentially for helping the chunking happen. Or maybe it's together, the VTA and substantia nigra work together to help chunk individual actions into one larger unit. I don't know that yet. So we're trying to find different ways to interrupt the function of one or both to see if we can reveal that dependence.
And then for the second part, related to this, when you stimulate dopamine neurons, the CS response developed in dopamine neurons. Did you see dissociation of the development of CS response in BTA or SNC by stimulating the different dopamine neurons?
We did not, but in that study, there aren't enough individual subjects in each group for me to say that with certainty. So I think it's still possible that there will be differences either in the way they develop or some of their characteristics, but we don't know yet. So I think that's something still to be discovered, and you should do that experiment. Yeah.
Thank you. And then the third question, the first part, the learning of the state-state association by dopamine neuron. So is it possible because of from the second part of the talk, so the first part of the behavior, also dopamine neurons probably develop this response cue, right? By this stimulation. So then, if the response to cue is necessary to elicit the behavior, and the state-state association might be happening in somewhere without dopamine neurons, and then just the dopamine neurons was necessary to elicit the
But I wonder if what you're saying, it sounds like what you're saying further experiments could test that by inhibiting the dopamine neurons that's either during the learning or the performance, or changing the way that you're asking the animal to perform. Is that?
Yeah, so part of so, and then the interpretation is so dopamine CS response was important, and then that could have only the value information, and then the state information
I
was
see.
already there, but that behavior wasn't elicited because of the lack of the CS response.
Yeah, I have to think about a way to test that.
Yeah.
So when you had the light come on and the rats that were trained on VTA stimulation versus SNC stimulation, they both had movement initially, but different types of movement. It looked like when you measured the onset of the activity that it went to full speed right away for the VTA stimulation.
Actually, that was substantia nigra.
Oh, it was the other way around.
Yeah.
And is that just because of the type of action being different?
That's a great question. I actually don't know the answer to that. I'm sure I saw these data, but I can't remember over days how that evolves in the different groups. So I would like to know a little bit more about that. But that's a good question, and I don't know the answer to that. So some things that have been very interesting to me to think about is how the eventual conditioned response might depend, or does it at all depend on any actions the animal might be making during acquisition. And so I've been thinking about things like, can we still do the same pairing but restrict the animal's movement, and then when you let him go, what would you see then? And maybe for substantia nigra, you might predict not much, or maybe you would predict it would be the same. I think there's sort of two camps on that. VTA, you might predict the animal still will have somehow ascribe some value to the cue, so you still will see something. But I think these are actually open questions.
Yeah, thanks for the nice talk. In your blocking paradigm with your Q, A, and X, I was wondering, so when you give the artificial stimulation, you can drive this pairing, right? But do you know the time window of when, if you do the stimulation outside of when reward should come, right? So like, do you know what the sort of probability distribution is if you give it earlier or later, and then it no longer acts like is a cue?
Yes. So Ronald tried to pair the activation of dopamine neurons as exactly as he could with when the animals are getting each of the individual drops. So it's far from perfect, but he tried to do his best to give a short burst, and I can't remember right now exactly the length of dopamine neurons, some fixed milliseconds before or after when the drop comes. I can't remember that exactly either, but he tried to really make sure that when the animal was ingesting the reward, there also was dopamine neuron activation at that time. And it always occurred at a fixed time after cue onset.
Right. So, but I'm wondering if it's kind of opens an interesting question though about timing of reward, right? Because there's a lot of, there's like how you pair it's like temporal credit
Yes.
assignment.
Yeah.
So how far back or forward can you go if you don't pair it perfectly with the reward?
Yeah.
Do you know if you can still cause that learning?
No, that's a great question, and I actually want someone to do that experiment, and I can't get anyone to do that parameterization. I really, you might predict if the cue onset and the dopamine neuron stimulation, which is even substituting for reward, occur at the exact same time, that you would get nothing,
Exactly.
right? But so far, no one wants to do that.
Is there any limitation to the amount of time after implant that you're able to study these in the brain, or any kind of projection of the implant?
Not that we've found thus far, but we've gone out many months, but we haven't really tracked that carefully to see what the changes over time might be, or when we might have reached a time when there's been too much stimulation and then maybe a cumulative toxicity or something like this. We haven't done anything careful about that. One control that people usually do in the lab is just at least check that each animal will still do ICSS to make sure that the basic sort of baseline level of functioning is still happening, but we haven't done that.
Last question.
Okay.
So the beauty of the experiment is optogenic stimulation alone can cause cue approaching behavior without any reward. I wonder whether this behavior interacts with natural hunger first. If you have different motivations on baseline, any change that they learn by dopamine stimulation, or instead, is it always cue approaching that this BPS stimulation is causing to learn?
Yeah, that is a great question. And we haven't done that. And I'm not sure, do you mean if we had hungry animals and did the same experiment, but the animal had no food available and they never associated that chamber with food?
Well, for instance,
Or if it was
you
a,
could have reward code and food code,
Mhm.
but then you do this in extinction. So you
Mhm.
don't keep reward
Mhm.
in this environment, but then you use dopamine stimulation in a very similar way as you did.
Mhm.
And then in the presence of these, they might approach to these pods instead of cues.
Mhm.
And so that's kind of one way to do this.
Yeah, I think that would be interesting. We haven't done that. I presume that by our stimulation, we're sort of biasing the group of animals to sign track to the cue, whereas normally not every animal would do that naturally. But I like the idea of pitting it against other sorts of naturally conditioned behaviors, which I think would be interesting behaviorally, but I actually would like to know what's happening in the brain at that time and to look at the responses to potentially different kinds of cues, the dopamine cue and the natural reward cue and things like that. It's a neat idea. Thank you.